Hello everyone, welcome to Immunology-Serology Review. We will be discussing some immunology concepts and some serological tests used for diagnosis. For our immunology and serology discussion for today, we will differentiate nonspecific and specific immunity, also the complements and the immune response. And for the serology part, we will discuss some of the conditions that are diagnosed by using serological tests like syphilis, CRP, AIDS, febrile antibody tests, and Lyme disease. Let's start with the definition of immunity. Immunity is the defense mechanism to protect the body against the invasion by non-self substances. So that means multicellular organisms like ourselves have the capability or the capacity to recognize and to tolerate everything that belongs to the self. And the opposition of this is that they can resist or remove anything that doesn't belong to the self, and we call these as non-self substances because these non-self substances may cause harm to the body. Now, it has some positive effects and negative effects. A positive effect is that it can give us natural resistance, and if ever we do get sick, it can help us in the recovery. And after recovery, it can give us acquired resistance. Negative effect is that it can cause the rejection of an organ transplant because these organ transplants coming from other individuals may be considered as non-self by the body. Now, there are two types of immunity that are recognized, and we will discuss these. We have natural immunity and acquired immunity. To differentiate natural from acquired immunity, first is that natural immunity is also known as an innate immunity, while acquired immunity is also known as an adaptive immunity. It's named innate immunity because it is present at birth, while acquired immunity or adaptive immunity is named as such because it is acquired or it is formed after antigen exposure or after an infection. The second characteristic is that in natural it is nonspecific, while in acquired it's specific. Nonspecific means that it has a standard response to all the antigen. Whatever the antigen is, they would have the same response to it. Unlike in acquired, it will have a specific response because it has selected cells that would do the job for it, and it has antibodies that are specific to a certain pathogen. So if there's pathogen A, there would be an antibody A, and if there's pathogen B, there would be an antibody B. So they are specific in their response. Also, there would be diverse response to the antigen. So it's different antigens, different response by the antibodies. Next is that in natural, it is not antigen dependent, while in acquired, it is antigen dependent. Natural immunity does not need an antigen to be present for it to work, while in acquired, it needs antigen presence and presentation for it to start working. In natural immunity, there is no immunologic memory, while in acquired immunity, there are lymphocytes with memory. In natural immunity, we call the immune cells as phagocytes, and they have no memory. Their response does not change even after repeated exposure. In acquired immunity, we have immune cells, the lymphocytes, with memory B and memory T cells. Now, these types of cells recall previous antigen exposure. That's why in repeated exposure, the response will become faster, they become stronger, and they become qualitatively different because they remember what antigen is present and how they were able to destroy this antigen so they can work faster and better. Next one, natural immunity has an immediate response, while acquired immunity has a delay between exposure and the response. So in natural immunity, the response is very fast. After exposure to an antigen within 30 to 60 minutes, these white blood cells or the natural immune cells can start attacking the antigens, while in acquired immunity, there is delay of about 3 to 5 days because that's how long the system takes for it to produce antibodies. And then for the natural, we have the first and second line of defenses, while in acquired, we have the third line of defense. And Among these three lines of defenses, the third line of defense is the highest line of defense. There are three lines of defenses: the first, second, and third line of defense. The first and the second belong to the natural immunity, while the third belongs to the acquired immunity. Under the first line of defense are several components. We have the external barriers, body secretions, and other factors. Under external barriers, for example, we have the intact skin. The skin prevents the entry of unwanted organisms or pathogens inside the body, while the mucous membranes and the cilia from the respiratory tract, for example, help in expelling foreign substances that were able to enter the body. Body secretions like tears, saliva, and sweat contain enzymes that are able to destroy the bacterial cell wall. Other factors like pH, for example, may give a low pH in the vaginal area, for example, that would enhance the normal flora growth. While body temperature, if there's an invading pathogen, the body temperature will increase. That would help in killing the organism. Now, the second and the third line of defense has some similarities, although the second line belongs to natural immunity and the third line belongs to the acquired immunity. One of the similarities is that both of them have the cellular components. Under the second line of defense, cellular components are the granulocytes, monocytes, and the macrophage. We also have the natural killer cells and the antigen presenting cells. The cellular elements on the third line of defense are the lymphocytes and the antigen presenting cells as well. Another common factor is that both of them have humoral components. Under the second line of defense, we have the cytokines and the complement, and on the third line of defense, we have again the cytokines and the complement with the addition of antibodies. Under the second line of defense, as mentioned earlier, we have the cellular and the humoral components. Under cellular components, we have the granulocytes, monocytes, tissue cells, and lymphocytes. When we say granulocytes, we are referring to the white blood cells with granules, and we have the neutrophils, eosinophils, and the basophils. Tissue cells refer to the immune cells that are located in different tissues, like the macrophage, mast cells, and the dendritic cells. The lymphocytes in the second line of defense are different from the lymphocytes mentioned in the third line of defense. The lymphocytes here are called the natural killer cells, the lymphokine-activated killer cells, and the antibody-dependent cytotoxic cells. So these are different lymphocytes. Under the humoral factors are different chemicals, and just to enumerate them are the acute phase reactants, beta-lysine, chemotactic factors, complement, interferon, lactoferrin, lactoperoxidase, lysozyme, properdin, and tumor necrosis factor. Each of these components will be briefly discussed in the succeeding slides. The three granulocytes are the neutrophils, eosinophils, and the basophils. The neutrophils are the basic or the principal leukocyte associated with phagocytosis and localized inflammatory response. The granules contain bactericidal enzymes that respond to chemotaxis. Eosinophils produce histamine for inflammation. They only have some phagocytic ability because they are mostly for killing parasites. The basophils are the least type of granulocyte. They have granules with histamine and heparin that are released during allergic reactions, giving them a role in hypersensitivity reaction. Another type of cell is the monocyte. The monocyte is a mononuclear cell that is involved in the phagocytosis process. They also present antigens to the lymphocytes, and they migrate to different tissues to become a macrophage. They respond to chemotaxins for defense. The third type of immune cells with the secondary line of defense are the tissue cells. These are cells located in different tissues. We have the mast cells, macrophage, and dendritic cells. Mast cells, also known as mastocyte or labrocyte, are connective tissue cells that are very similar to basophils. One difference, though, is that the mast cell granules are larger and are more abundant. Both granules from the mast cells and basophils produce histamine and heparin during allergic or hypersensitivity reaction. The second type of tissue cells are the macrophage. These are types of cells that have differentiated from monocytes. The macrophage function in the elimination of bacteria by phagocytosis. They can also present antigens on their cell surfaces. The names of the macrophage will vary depending on where they are located. Alveolar macrophage are located in the lungs, histiocytes in the connective tissue, Kupffer cells in the liver, mesangial cells in kidneys, microglial cells in the brain, osteoclasts in the bone, synovial A cells in the joints, splenic macrophage, spleen, Langerhans cells in the skin and mucosa, and the Hofbauer cells in the placenta. The third type of tissue cell are the dendritic cells. A special kind of dendritic cell called the Langerhans cells are present in all layers of the epidermis and are most prominent in the stratum spinosum. The main function of dendritic cells is to process the antigenic material and present it on their cell surfaces for the T cells of the immune system. Also, they are able to perform phagocytosis. The dendritic cells are considered as the bridge in between innate immunity and acquired immunity, as they initiate the start of the acquired immune response. The last type of cells in the second line of defense are the lymphocytes, the non-T and the non-B lymphocytes. Again, these are lymphocytes but not the T lymphocytes and the B lymphocytes that we know or that are involved in the third line of defense. The first non-T, non-B lymphocyte of the second line of defense is the natural killer cells or the NK cells. These cells are also known as null cells or third population lymphocytes. They are large lymphocytes with granules. They are considered as the first line of defense against tumor cells and virally infected cells, and they bridge the gap between the innate and the acquired immunity. The second type of non-T and non-B lymphocytes are the lymphokine-activated killer cells. Like the natural killer cells, they have the same function for killing cancer cells and for lysing tumor cells. LAC cells are actually natural killer cells that have been exposed to a lymphokine called the interleukin-2, giving them a heightened toxicity or cytotoxicity toward malignant cells. Hence the name lymphokine-activated killer cells. Again, LAC cells are natural killer cells that have been activated after exposure to a lymphokine called interleukin-2. The last type of non-T and non-B lymphocytes would be the antibody-dependent cytotoxic cell or the ADCC cell. These cells are also known as the K-type lymphocyte. These ADCC cells requires an effector cell or cytolytic effector cells. Examples of cytolytic effector cells would be the macrophage, natural killer cells, eosinophils, and neutrophils. ADCC cells are able to lyse target cells without the complement, and they can also lyse antibody-coded target cells if there is direct contact. We have now discussed all the cellular components of the second line of defense. Let's now continue with the humoral components. The first component of the humoral second line of defense are the acute phase reactants. Acute phase reactants increase during the time of inflammation, hence the name. They get to react in the acute phase of inflammation. Their reaction is to increase in concentration or amount. An example of acute phase reactants is CRP. CRP is the most common acute phase reactant. It activates the complement classical pathway, and it can increase 20 to 1,000 times during inflammation. The second type is the beta-lysine. The beta-lysine is a protein that is released from the platelets when they rupture. They rupture during clot formation. Beta-lysine is primarily active against gram-positive bacteria. The third factor are chemotactic factors. Now, chemotactic factors don't have a direct attack against pathogenic organisms, but what they do is that they stimulate other organisms, other immune cells, to go to the site of infection or to go to the site where there is a pathogen. Now, chemotactic factors may either be chemotaxins or chemokines. Chemotaxins source may either be endogenous or exogenous. An example of endogenous source would be a C5a, which is a byproduct of the complement. So when C5a is formed during the complement activation, it can act as a chemotactic factor, also calling the other immune cells. Example of exogenous chemotaxins are dead cells or necrotaxins. Now, when dead cells are seen floating around in the bloodstream, these can also signal the other immune cells to come. Chemokines are cytokines. They are a family of small cytokines, and when we say cytokines, they are derived from the ability to induce direct chemotaxis in nearby cells. The fourth one is the complement. The complement is a series of over 30 proteins whose main function is to be able to mediate inflammation. Their mediation is to enhance the ability of antibodies and phagocytic cells to clear the microbes and damaged cells. They are able to attack the cell membrane of the pathogen, therefore causing its lysis. The complement series are synthesized in the liver, except for C1, which is produced by intestinal epithelial cells. The precursors or inactive precursors are called zymogens, and these zymogens will be converted into active enzymes. The major functions of complement are inflammatory, opsonization, and cytotoxicity. For inflammatory, the activation of the complement will cause the release of histamine by the mast cells and the basophils, thus stimulating the inflammatory response. For opsonization, the complement facilitates phagocytosis by coating the pathogenic organisms or immune complexes. This opsonization makes the pathogenic organism more attractive or easily eaten by our phagocytes. For cytotoxicity, this is the final stage of the complement cascade, where the membrane of the pathogen is attacked, therefore leading to cell death and cell lysis. There are three pathways that can activate the complement system, and these are the classical, alternative, and the lectin pathway. Complement components of the classical pathway starts with C1, C4, C2, and C3, leading up to C5 to C9, which is also known as the membrane attack unit. The alternative pathway skips C1, C4, and C2 of the classical pathway, but rather starts at C3 and going to C5 to C9 for the membrane attack unit. The lectin pathway has no C1. It starts at C4 and continues on like the classical pathway until it gets to the membrane attack unit of C5 to C9. The fourth humoral component is interferon. Now, these are glycoproteins that are antiviral in activity. They are called interferon because they are able to interfere with the viral replication, therefore stopping its processes and multiplication. There are two types of interferon. We have type 1 and type 2. Under type 1, it can be interferon alpha and interferon beta, and type 2 has interferon gamma. Interferon alpha are produced by NK cells, therefore called as leukocyte interferons. Interferon beta are produced by epithelial cells and fibroblasts, therefore called fibroepithelial interferons. And interferon gamma are produced by T cells, also known as immune interferons. The next one is lactoferrin, which is previously known as lactotransferrin. Lactoferrin is a protein that is found in milk that binds with iron, therefore competing with bacteria or other pathogens. Lactoferrin is considered to be antibacterial, antiviral, and antiparasitic in activity. Lactoferrin is found highest in amounts in colostrum, which is the first type of breast milk found after a baby is born. The next one is lactoperoxidase, which is a peroxidase enzyme, which is found in saliva and milk. Lactoperoxidase has the same microbicidal effect as myeloperoxidase. This is also considered as a natural antibacterial. The last of the humoral components are lysozyme. Lysozyme is present in tears, saliva, nasal secretions, and other body fluids. These contain lysozyme that destroys the bacteria that attempt to enter our body. In the case of tears, they protect our eyes from bacterial invaders by hydrolyzing the cell wall of the bacteria. The next one is properdin. Properdin is a protein that acts to stabilize the alternative pathway. It enhances the alternative pathway, therefore being antibacterial and antiviral. And the last one is tumor necrosis factor, or TNF. This is a type of cytokine, so that means it gives signals to other cells, which is cytotoxic to tumor cells and virally infected cells. Therefore the name tumor necrosis factor. There is TNF alpha, which is produced by the macrophage, and TNF beta, which is produced by CD4 and CD8 cells. Before we go to the third line of defense, let's first discuss some processes of the innate immune system. The first process is phagocytosis. Phagocytosis is defined as the ingestion of other cells or substances by phagocytes. It has different stages. There are 4 stages that makes up phagocytosis. The first stage is initiation, which is due to the tissue damage or the presence of microbial agents. The second stage is diapedesis or chemotaxis. These two will be differentiated in the following slide. But if there is an abnormality in any of these two, we can have disorders like Jobs syndrome and lazy leukocyte syndrome. In Jobs syndrome, there is an abnormal chemotactic activity. But in the lazy leukocyte syndrome, there is an abnormal random and chemotactic activity. The third stage is engulfment, where there is the formation of the phagosome by the active amoeboid movement, and opsonins are able to interact with the surface of the bacteria that makes them acceptable for phagocytosis. This process is called as opsonization. The last stage is digestion, where there is the formation of the phagolysosome that leads to the digestion of the foreign materials. Chemotaxis is the movement of phagocytes towards or away the pathogen as guided by the chemotaxins. Chemoattractants attract phagocytes towards the pathogen, while chemorepellents push away the phagocytes away from the pathogen. Diapedesis is the migration of the phagocytes from the blood vessels going to the tissues. So this is when the white blood cells flowing in the blood vessels would be attracted to the chemoattractant and would try to squeeze themselves in between the capillary blood vessel so that they can go inside the tissues where the pathogen is. We are now done with the innate immune system. Let's now go to the third line of defense, which is part of the acquired immune system. The third line of defense also has cellular and humoral components. Under cellular, we have the lymphocytes and antigen presenting cells, while in humoral, we have the cytokines, complement, and the addition of the antibodies. The two lymphocytes in the acquired immune system are the T cells and the B cells. Both cells are from the bone marrow, but the T cells have developed their specific surface antigen in the thymus, therefore called as the T cells. T cells are 60 to 70% of the lymphocyte population, while B cells are 10 to 20%. T cells are cell-mediated, while B cells are humorally mediated. T cell products are cytotoxins like perforins and granzymes, while the B cell products are antibodies. T cells are more on antiviral, antifungal, anti-tumor, and are responsible for graft rejections, while B cells are for bacterial infections. T cell surface markers are CD2, 3, 4, 5, 7, and 8, while B cell surface markers are CD19, 20, 21, and 22. Receptors for T cells are called T cell receptors, while B cells are surface immunoglobulin G receptors. The mitogens that can be used for T cells are phytohemagglutinin, concanavalin A, pokeweed, while in B cells, pokeweed may also be used with the addition of staphylococcal protein A. There are two subsets for T cells. We have the helper T cells and the cytotoxic T cells. Other names for helper T cells are inducer T cells, and cytotoxic T cells are also known as regulatory T cells or Tregs for short. Let's start with the helper T cells. As the name implies, helper T cells helps in cell-mediated immunity. They may be the most important cells in the adaptive immunity because they are required for almost all adaptive immune responses. For example, they help activate B cells to produce antibodies. They also help macrophage to destroy ingested microbes. And they are also needed for T cells and natural killer cells to kill infected cells. They recognize class II MHC molecules. Other name of helper T cell is also CD4 cell, and these CD4 cells or helper T cells are the main target for HIV. That's why a decreased amount of CD4 cells may be seen in AIDS. The next subset is the cytotoxic T cell. These were formerly known as suppressor T cells. Their function is to suppress or to inhibit T helper cells from becoming overactive. They are immunosuppressive. Also, they are called cytotoxic T cells because they can kill other cells like tumors or cells that were virally infected. Another name for cytotoxic T cell is CD8 cells. Now, since CD4 cells are affected by HIV, a decreased amount of CD4 cell will also affect the quality of CD8 cells. So in HIV conditions, CD8 cells may dysfunction. The second type of lymphocyte are the B cells. B cells, when activated, are mature B cells. They can be identified by surface immunoglobulins. Their end product is the production of antibodies. Now, after antigen exposure with the aid of helper T cells, they will transform into blasts that can become either plasma cells or memory B cells. When they become memory B cells, they can respond to reoccurring antigen. So these are B cells that have the memory or that can remember which type of antigens have attacked. So when this same antigen occurs or reoccurs in the body, they can attack in increased speed and intensity, and they can live for a long time, months up to years. When mature B cells differentiate into plasma cells, the plasma cells will be the ones to produce antibodies. That is their main function for the production of antibodies. And unlike memory B cells, plasma cells will die in a few days after antibody production. The next cells in the acquired or adaptive immunity are the antigen presenting cells. Now, as the name implies, these types of cells have the capability to present antigens on their surfaces. And we will have three cells. These are the dendritic cells, the macrophage, and the B cells. So that means all of these three cells are able to showcase the antigen like little flags on their surfaces, so that other immune cells that have not encountered the antigen yet will be warned that there is a pathogen in the surrounding and that they should keep a lookout for them. So the first one is the dendritic cell. Now, dendritic cells are also known as accessory cells. Their main function is to process the antigen material and to present it on the cell surface so that T cells of the immune cells will be warned. Now, they act as messengers between the innate and the adaptive immune system. If you remember, dendritic cells are also a part of the innate immunity. Now, after processing these antigen materials, these antigens would be placed on the surface of the dendritic cells with the help of the MHC class 1 or class 2 molecules. Next would be the macrophage. So after phagocytosis of the antigen, parts of the antigen will be attached to the MHC 1 or 2 molecules, and these will also be placed at the surface of the cells. And they will be shown to different cells, and the other cells will see that these types of antigens are present and that they should attack. These cells do not phagocytize, but they are able to express the antigen on their cell surfaces as well, which are associated with the MHC class 1 molecules. The MHC class 1 molecules can bind to CD8 cells or to cytotoxic T cells. So when these cytotoxic T cells that have not yet encountered the antigen will cross paths with the antigen presenting cells, they will be warned that there are these types of antigens. This is how the antigen looks like. Go and look for it, kill it. And MHC class 2 molecules can bind to CD4 helper cells. So when the helper cells sees these flags or antigens presented on the cells of these types of cell, it can signal them to help other cells. They can help B cells, they can help other macrophages who are attacking, and so on. Now, the heightened immune response is caused or helped by these antigen presenting cells, so that the elimination of pathogens will be faster. We have mentioned the major histocompatibility complex earlier, or the MHC, so let's discuss them a little bit more. So MHCs are groups of genes that are found in chromosome 6 that code for proteins found in the surfaces of cells that can help in the immune system recognizing foreign substances, as what we have explained earlier. Now, the MHC proteins are found in all higher vertebrates. In humans, the products are called as human leukocyte antigens, or HLA. One clinical significance for MHCs or HLAs are for paternity testing, where the HLAs are determined for histocompatibility. MHCs have 3 classes: class 1, class 2, and class 3. Class 1 MHCs are glycoproteins expressed in all nucleated cells. Their major function is to present antigens to cytotoxic T cells. Now, these class 1 are found in genes in the regions of HLA A, B, and C. Class 2 molecules are expressed in B cells and all antigen-presenting cells. Their genetic loci are found in the regions of HLA DP, DQ, and DR. Their major function is to present antigens to helper T cells. Now, class 3 molecules, unlike class 1 and class 2 that are well defined, class 3 are poorly defined structurally and functionally. What we know is that they have products that include proteins that have immune function. For example, in the complement components, we have C2 and C4. They also are involved in TNF and inflammatory molecules. Just to reiterate, HLAs, or human leukocyte antigens, are different from MHCs. HLAs are the group of proteins encoded by the MHC in humans. HLAs are the markers found on the surfaces of each of the cells. HLAs are responsible for the regulation of the immune system by allowing the immune systems to be able to distinguish self from non-self. Since the HLAs are unique for every person, the immune system would be able to tell if you belong to that specific person. So an individual's HLA will be different from another person's HLA. They serve more like of a fingerprint, but for every cells in the body. That's how the immune cells know if you do belong to the self or if not. If you have a different set of HLA, you will be considered as a non-self, and you will be considered as a threat or as a pathogen. That is why in transplants, HLAs should be modulated. For example, if the HLA of the patient is very different from the HLA of a recipient, the transplantation will not be successful. So it is important that the HLA of the patient and the recipient are matched as closely as possible so that the transplants can last longer. For platelet transfusion, the primary cause of immune-mediated platelet transfusion is the antibody to the class I HLA antigen. In paternity testing, it is based on the HLA inheritance patterns that can exclude the fathers with approximately 99% because the HLA loci are polymorphic and that the recombination is very rare. The HLA of the father would be very much the same, or if not 100% identical, to the HLA of the offspring. So then they can identify if the person is the father or not of a person. For the diseases, we have to remember that not all the individuals who have a particular HLA will have the disease, but many individuals with certain diseases do express a particular HLA. For example, individuals with rheumatoid arthritis have the HLA-DR4 in their cells, while patients with ankylosing spondylitis have the HLA-B27. So in short, the HLAs do not cause the disease, but they do serve as markers for the presence of the disease. We are now done with the cellular components of the third line of defense of the acquired immune system. Let's now go to the humoral components. For the humoral factor in the third line of defense, we have 3: cytokines, complement, and antibodies. The complements have been described earlier, starting from C1 to the membrane attack unit that leads to the lysis of the cells. And there are 5 types of antibodies produced by plasma cells. We have immunoglobulin G, A, M, D, and E. And these 5 antibodies will be differentiated later on. But first, let's talk about cytokines. Cytokines are signaling proteins. They are protein hormones that are secreted by cells. Their function is to stimulate or to inhibit antigenic response. Examples of cytokines are interleukins, colony-stimulating factors, tumor necrosis factor, interferons, and others. Interleukins are a group of cytokines that have more than 50 different types. They come from different sources and they all have different types of actions. Interleukins are chemical messengers that serve to regulate the immune system by sending messages both on the innate and the acquired immune response for infections. The word interleukin comes from 2 words, inter and leukin. Inter meaning communication because they serve as messengers, and leukin because of the old fact that many of them are produced by leukocytes and that they act as messengers in between other leukocytes. But nowadays, we know that there are different sources of interleukins, and the term interleukin just remained. Lymphokines are interleukins that are produced by lymphocytes. Examples of interleukins are interleukin-1 alpha. This is coming from the macrophages and the monocyte. What they do is that they send signal to the hypothalamus to cause a fever. So when there's a pathogen present, the macrophages and the monocytes produce interleukin that acts on the hypothalamus so that there would be an increase in the body temperature that would make the environment not suitable for the pathogen. Another interleukin is interleukin-2. The source of this is the T cells, and they stimulate the LAC cells or the lymphokine-activated killer cells, which is part of the innate immunity. Interleukin-7, its source is the bone marrow, and it's responsible for the proliferation of pre-B, pre-T, and the activation of mature T cells. The last example is interleukin-12, which source is also the T cells, but its role is to activate natural killer cells. 2 more cytokines are TNF and IF, or tumor necrosis factor, and interferon, which are also a part of the humoral second line of defense. Just to give more information, TNF activates the immune system to help kill cancer cells, and these are produced mainly by the macrophages. There are two types of TNF, TNF alpha and TNF beta. TNF alpha is also known as cachectin and is considered as an acute phase reactant because it can cause local inflammation, leading immune defenses to protect a localized area. TNF beta comes from T and B cells, and they are considered as lymphotoxin, meaning that they can kill target tumor cells. Interferons are antiviral. They are a group of signaling proteins that can warn cells around them. So they are produced by infected cells, virally infected cells, so that they can tell the other healthy cells around them that they have been infected. Interferon alpha are produced by leukocytes, interferon beta are produced by infected fibroblasts, and interferon gamma are produced by infected T cells and infected natural killer cells. This is an example of one interferon, interferon alpha, produced by neutrophils. So when a virus infects a neutrophil, the neutrophil will start producing interferons, and these interferons will warn the surrounding neutrophils about the danger. And when they are warned, they will start to produce antivirals. So when the virus starts to infect the surrounding neutrophils, they are ready to attack the virus so that the viral replication will stop and will not infect the neutrophils. The last humoral component of the third-line defenses are the antibodies, also known as immunoglobulins. So these are substances produced by plasma cells in response to an antigen. So they will give a specific interaction, meaning that they will only react to the antigen that they were made for. These are the major humoral components of the adaptive immunity. Its function is to neutralize the toxic substances, facilitate phagocytosis, and to combine with antigens on the cell surfaces for cell destruction. This is the structure of an immunoglobulin. It is a tetrapeptide structure, meaning it has 2 heavy chains and 2 light chains. It has 2 types: the variable antigen binding site, where the antigens attach, and the constant carboxy terminal end, where other immune cells attach. CH2, or the second heavy chain, is the receptor for complement fixation, where the complements can bind. CH3, or the third heavy chain, is the receptor for phagocytes for phagocytosis. The hinge region, which is the flexible portion of the antibody, is responsible for the flexibility of the arms of the antibody. This is located in between the heavy chain 1 and heavy chain 2. And lastly, the heavy chains and the light chains are held together by disulfide bonds. Antibodies can be divided into different fragments by using proteolytic enzymes. These enzymes can cut the peptide bonds of the antibodies. They will degrade the immunoglobulins into definable fragments. We can use 2 enzymes, papain and pepsin. Papain divides the antibody into 3 fragments. That would be 2 Fab arms and 1 Fc fragment. Pepsin, when used, can divide the antibody into 2 fragments, which is cut below the hinge region. So we will have 1 Fc fragment and 1 fragment that has the 2 arms linked together. There are 5 different classes of antibodies. We have IgG, IgM, IgA, IgD, and IgE. For the structure, only the IgM is found to be a pentamer. Pentamer because it has the 5 basic structural units in a circular arrangement. IgA may also be found as a dimer, while the rest are found as monomers. For the percent, IgG is the one that is found the most abundant, followed by IgA, and the least abundant is IgE. For complement fixation, only 2 of them can bind to complements, and these are IgG and IgM. The rest cannot, although IgA may only alternatively bind. And a unique characteristic of IgG is that it can cross the placenta. The other 4 antibody classes cannot. IgG has 4 subclasses: IgG 1, 2, 3, and 4. IgG 1 and 2 are related to placental crossing, and IgG 3 and 4 are concerned with complement fixation. Between IgG 1 and 2, IgG 1 is most efficient in placental crossing. It is also the major subclass of IgG. And IgG3 and 4, between IgG3 and 4, IgG3 is the most efficient in complement fixation, while IgG4 is the least efficient in complement fixation. IgM is considered as the most efficient out of all the antibody classes in complement fixation and agglutination because of its size. IgA has two subclasses, IgA1 and IgA2. IgA1 is primarily appearing as a monomer in serum, while IgA2 is found as a dimer in body secretions. IgD is an immunoregulation. It is considered as an antibody that is against an antibody, meaning it is involved in a feedback mechanism. It switches off B cells so that it can stop when it is not needed anymore. IgE is a reagenic antibody. It attaches to surfaces of mast cells for mast cells to release granules. This happens during the times of type 1 hypersensitivity reaction. There are 4 phases in antibody production or antibody synthesis, and these are lag, log, plateau, and decline. The first stage is the lag phase. This is when the pathogens have entered the body, so there are no antigen production happening in these days. These are usually 3 to 5 days of no antibody production. The next stage is the log stage, also known as the exponential phase, where there is an increased antibody production. So this is when the B cells have already been differentiated into plasma cells, and the plasma cells have started the production of antibodies. The third stage is the plateau stage. The plateau stage is described as an equilibrium between the antibody synthesis and antibody catabolism. So here there is production and there is decline of antibodies. And the last phase is the decline phase. This is when there is an increased antibody degradation. This is when catabolism already exceeds the production of antibodies. An immune response or an antibody production may either be the primary antibody production or primary immune response, or a secondary immune response or secondary antibody production. Look at the image below as I differentiate each. In the primary immune response, this is when the exposure to the antigen happens for the first time, while in the secondary antibody production, this is the repeated infection from the same antigen. The primary antibody production has a longer latent phase. Latent phase is also known as inductive or lag phase. It happens for 5 to 7 days, while in the secondary antibody production, it has a shorter latent phase at 3 to 5 days. Plasma production happens in the primary antibody production, while antibody production is more concentrated and faster during the secondary antibody production. The antibody produced during the primary immune response is the IgM, while in the secondary immune response, it is the IgG. This is because of the class switching that happens in the genes. In the primary antibody production, there is a slow antibody production, while in the secondary antibody production, the production of the antibodies themselves persists longer. Let's now discuss the different types of adaptive immunity. There are 2 types of humoral-mediated adaptive immunity, and these are active and passive immunity. In active immunity, it requires the exposure to the antigen, so this antigen will then cause the production of antibodies by the host. Active immunity may either be natural or artificial. Natural is when the person is infected with an organism, while artificial is when the person is exposed to an antigen due to vaccine. In passive immunity, this is when the person is not exposed to an antigen. There is no antigen exposure. So the antibodies are not produced by the host, but they come from somewhere else. It can also be natural or artificial. In natural passive immunity, the antibodies are naturally produced by other individuals, like in transplacental antibodies or in breast milk. While in artificial immunity, artificial passive immunity, the antibodies are produced by another individual that is, for example, used in immune therapy. So that ends the immunology part of the discussion. Have a break first, and then proceed to the serology discussion. Thank you.